Hello, this is Tommy. Welcome back to Genomics. In today's video, I'm going to show you how to use annotation packages in R to get information such as the total length of the exons for each gene, or the total length of a gene, including the gene body, the introns. How do you get those information? Why do you need those information? Because, for example, when you are calculating RPKM normalization to actually calculate the total length of exons for each gene, right? Or if you are actually normalizing ChIP-seq data which occur in the whole gene body, such as H3K36 trimethylation, you need to calculate the length of the whole gene. Use that as a normalization factor. So I'm going to show you how to use annotation packages in R, bioconductor packages, and how to do that practically. So by the way, I will have the script in the description of this video so you can download and then follow along. So let's first load these two libraries. So in this case, I'm going to use the mouse genome, but you can also download the corresponding human databases. For example, I'm actually loading the library. So if you don't have those two packages, you need to use BioC Manager to install them. Okay, I'm using this transcription database and this OrgDB mouse. So once you load these two libraries, actually you have these two objects actually in your environment. So this will be a TXDB transcript database, and this will be just OrgDB organism database. Okay, so for humans will be for example, MM will be like HS, for example. So you can just search in the bioconductor page. Okay, so once you have that, so I'm just assigning this long thing to this shorter name TXDB. So once you have this transcript database, you can use this function called genes TXDB, and it will return all the genes in this database. Okay, so if you do this, and okay, there's a warning here. So 84 genes actually will drop because they have exons located in both strands of the same reference sequence or more than one reference sequence, or cannot be represented by a single genomic range. Actually, this is a little bit more detailed information. For some genes, actually, it's kind of strange. For example, some genes may have the start in one chromosome and the end in another chromosome. I mean, the same gene name may appear actually in different chromosomes. Actually, I saw one of the posts in the BioStars, and then there are such actually genes exist in the genome. Okay, so let's take a look how it looks like. So essentially, this is just a genomic ranges object. So each row here will be one gene. So this is untrans ID, and this is the start and end for that gene. So by default, this is actually it's taking the longest gene, the start, because a single gene can have multiple transcripts, and it's actually taking the longest transcript as the start and end. Okay, so this gene ID, and you might want to actually convert them or add another column for gene symbol. So what you can do, you can use this organism DB and use the annotation DBI package, use the select function, this is the database, and the key will be this column. So the gene ID, untrans, and the columns is like which column you want to map to. This will be the symbol, the official gene symbol, and the key type. This is untrans ID. So if you do that, it says, okay, return the one-to-one mapping between keys and columns. You can take a look. So you have one column for untrans ID and one column for the symbol. So you can just make sure they're all equal. So this will be just this column, and gene symbol is this data frame, and access this untrans ID. Then just make sure this is the same as this. Okay. So it says true. So now we are safe to add that symbol into here. Okay. So create a new column in here. It's called symbol, and put that symbol column into the genomic ranges object. If you take a look now, Now, actually, just take a look at this. Now, like for this genomic ranges object, now you have two columns, like in the metadata column, gene ID and a symbol. Okay, and we want to calculate the total length, right? In R, in bioconductor genomic ranges object, you can just use the width function, and it will give you the length of each genomic ranges. Now we can create a data frame. So we have gene ID, have the gene symbol, and then the last column will be just the length of that gene. So remember, that's the longest transcript. So if you do that, make a data frame, and now you have a data frame like this. So you can get the longest actual gene length for each gene in this case. Then how are you going to get the total length of exons for each gene? So you need to use this function called exons by txtb, and then you say by gene. Okay, let's see how it looks like exons. Actually, exons, this will be actually genomic range list object. Okay, and this is essentially okay by gene. So you are actually grouping the exons for each gene. So here, this is the unchanged ID for that gene, and this is all exons for that gene. Okay, because you have multiple exons for the same gene, right? And this is actually for the second gene, and then now you have a bunch of exons here. Okay, so what we can do, you can still use the width function, and it can. So in R, this is why you need to use those built-in functions, because this actually is vectorized. Even it's a genomic ranges list object, it can actually work on that. So if you do this with exons, now you get this integer list. Each one will be one gene, still the unchanged ID, but each number here is the length of that exon. So essentially, for example, for the first gene, you have 1, 2, 3, 4, 5 exons, and then you have 5 numbers. And to calculate the total length, you just need to sum them up. Like for example, now you just need to do the sum. And again, this is also vectorized. It's actually magic in R. So if you just use this, and then give this as the input, and let's just take a look at the first 6. So now you get this number. Okay, this is the total number of bases for the total exons for that gene. This is unchanged, and this is essentially what you have, right? You can actually, this is just a named vector, so the values are the total length of the exon in bases, and this is just the unchanged ID. So you can use the table function to make it as a data frame. So the name will be unchanged ID, the value will be the exon length. So let's take a look. And if you look at this data frame, now you have the entrance ID, then you have the total exon length for that gene, right? This is very, very useful. Of course, you can add another column, just like what we did for the gene symbol. Okay, I hope this video is helpful for you, and make sure you get familiar with all the annotation packages in R, and it will make your life much easier. So instead of actually going to NCBI or UCAC to get all those annotations like manually, like bioconductor is a much better place for you to just use that package, and everything will be in that database in our object in your system. Okay, that's it for today. I will see you next time, and make sure you subscribe. See you next time. Bye. Happy learning.